Hello, dear students, welcome to our program. Today we are going to discuss bacterial transformation, transduction, in which we will see generalized and specialized transduction. The main objectives of today's topic are to have a general introduction of the process of transformation, to have a detailed account of the mechanism of transformation, to discuss the mapping of genes by transformation, to have a general introduction of the process of transduction, to discuss generalized transduction, to discuss specialized transduction, and to have a thorough idea about the co-transduction. Before we start, let's discuss horizontal gene transfer. Horizontal gene transfer is common among bacteria, even among very distantly related ones. This process is thought to be a significant cause of increased drug resistance when one bacterial cell acquires resistance and the resistance genes are transferred to other species. Horizontally transferred genetic elements play important roles in the virulence of E. coli, Salmonella, Streptococcus, and Clostridium perfringens. Transformation is one of the three forms of horizontal gene transfer that occur in the nature among bacteria, in which the DNA encoding for the trait passes from the one bacterium to another and is integrated into the recipient genome by homologous recombination. Transduction carried out by the means of bacteriophage is another mode of horizontal gene transfer. Now let's move to the process of transformation and discuss it in detail. Transformation is a mechanism of genetic transfer between bacteria in which the donor DNA is cell-free DNA in the recipient bacterium's immediate environment. DNA can be naturally released into the environment when cells die and recipient lyses. Once the free DNA comes in contact with a competent bacteria, linear double-stranded DNA enters into the cell, but one strand is degraded while the other is integrated into the chromosome by homologous recombination. Recipient cells that undergo this process and acquire a new phenotype as a result are said to be transformed. Single-stranded exogenotes are unstable and will usually be degraded unless they are integrated into endogenote. By the process of homologous recombination, the transforming DNA integrates into the bacterial chromosome. If the exogenote contains an allele of the endogenote, the resulting recombinant double helix would contain one or more mismatched base pairs and is referred to as a heteroduplex, that is, double-stranded DNA in which the two strands have different origins. These mismatches are repaired by DNA repair enzymes that scan the DNA and repair. The enzyme systems preferentially replace the base in the new strand, thus restoring the genotype of the recipient. If the region is replicated before it's repaired, however, one of the two daughter chromosomes will be recombinant and the other will be identical to the recipient. Dear students, there is an important event known as mapping by transformation. Transformation is used for mapping. Mapping by transformation experiments is based on the principle that two genes transform together if they are near enough to be carried on the same DNA fragment. Let's consider two genes, A and B, present on the bacterial chromosome. If these genes are widely separated in the chromosome, that they are always carried on two different DNA fragments, the probability of simultaneous transformations of the A-B recipient is very less. If the two genes, however, are so near to one another that they are often present on a single DNA fragment, the frequency of simultaneous transformations is same as the frequency of single gene transformation. Thus, the frequency of co-transformation is inversely proportional to the distance between two genes. Dear students, let's now discuss bacterial transduction. In transduction, bacteriophages function as vectors to transfer DNA from donor bacteria into the recipient bacteria. The transfer of bacterial genes by the phage was discovered by Lederberg and Zinder in 1951 in Salmonella typhimurium. There are two types of transduction, generalized and specialized. Let's first discuss generalized transduction in detail. In this type, transducing phages produced during lytic growth are aberrant and contain a random fragment of the bacterial genome instead of phage DNA. Each individual transducing phage carries a different set of closely linked genes representing a small segment of bacterial genome. So in generalized transduction, each part of the bacterial genome has approximately the same probability of being transferred from donor to the recipient bacteria. When a generalized transducing phage infects a recipient cell, expression of the transformer donor genes occurs. Typically, phage that can mediate generalized transduction include P1, that's E. coli, and P22, that's Salmonella. The generalized transduction may be abortive or complete. Abortive transduction refers to the transient expression of one or more donor genes without formation of recombinant progeny, whereas complete transduction is characterized by production of stable recombinants that inherit the donor genes and retain the ability to express them. The DNA remains double-stranded during transfer and both strands are integrated into the endogene node. In abortive transduction, the donor DNA fragment does not integrate with endogene node and also does not replicate. And among the progeny of the original transductant, only one bacterium contains the donor DNA fragment. The frequency of abortive transduction is typically 1 to 2 orders of magnitude greater than frequency of complete transduction, indicating that most cells infected by generalized transducing phages do not produce recombinant progeny. Now let's discuss specialized transduction in detail. This transduction is mediated only by specific temperate phages, and only a few specific donor genes can be transferred to recipient bacteria. Specialized transducing phages are formed only when lysogenic donor bacteria enter the lytic cycle and release phage progeny. There are two distinguishing characteristics of specialized transduction. First, the only bacterial genes that can be transduced are those very near the site at which the prophage is integrated. For example, the only site at which lambda phage integrates into the host chromosome is between the genes for the galactose fermentation GAL and biotin synthesis BIO. So if the prophage disintegrates abnormally from the host chromosome, only the GAL or BIO genes could be transduced. Second, it results from the defective excision of the prophage from the host chromosome. The specialized transducing phages contain both host and phage DNA linked in one single DNA molecule. They are rare recombinants which lack part of the normal phage genome and contain part of the bacterial chromosome located adjacent to the prophage attachment site. Formation of the lambda GAL and lambda BIO transducing particles cause loss of some lambda genes. Lambda GAL particles lack the tail genes and sometimes head genes, both of which are located at the right end of the prophage. The lambda BIO particle lacks genes from the left end of the prophage. The number of the missing phage genes depends on the position of the cuts that generated the particle. The head and the tail genes are essential, so lambda GAL transducing particles are unable to perform lytic cycles and thus plaques. They are defective and this is denoted lambda D-gal. The genes missing in lambda BIO transducing particles are not essential for the lytic growth, so lambda BIO phage are usually plaque forming and are called lambda P-BIO, in which the P stands for plaque forming. The lambda D-gal transducing particles lack phage genes required to grow lytically. Then how are they formed? The transducing particle is produced by aberrant excision from a normal prophage. The prophage contains all of the essential genes, and hence the necessary gene products, that's head and tail proteins, are still present in the chromosome. Initially producing the defective transducing particles is not a problem, but there is a deficiency of essential gene products when the transducing particle infects a new cell. If a bacterial cell is double infected with a wild-type lambda phage and a lambda D gal phage, the wild-type phage can supply the functions of the missing in the defective phage, and the progeny will contain about equal numbers of both types. Specialized transducing phage are formed by aberrant excision of a lambda lysogen. This is a very rare event as compared to the normal excision via recombination between attenuated L and attenuated R. When the aberrant excision initially occurs, it will yield less than one specialized transducing phage per 10 lakh wild-type phage. Thus, this lysate is called a low-frequency transducing lysate, or LFT. If this specialized transducing phage co-infects a new host together with a wild-type phage, a dilysogen can result. Induction of the dilysogen yields a lysate containing 50% specialized transducing phage and 50% wild-type phage. Thus, this lysate is called a high-frequency transducing lysate, or HFT. Dear students, now there is another thing, co-transduction. We'll discuss it in detail. During transduction, there is a maximum size of the DNA segment that can be packaged by the bacteriophages. If two genes are close enough along the chromosome, a bacteriophage may package a single piece of the chromosome that carries both genes and transfer that piece to another bacterium. This phenomena is called co-transduction. The likelihood that two genes will be co-transduced depends on how close they lie. If two genes are apart along the bacterial chromosome, they will never be co-transduced because the bacteriophage cannot physically package a DNA fragment that's large. The co-transduction can be used to map genes present in the bacterial chromosome. Mapping of genes by process of co-transduction is based on the fact that the closer the genes to each other, greater the probability of their co-transduction. That is, the frequency of co-transduction is inversely proportional to the distance between the two genes. For example, consider three genes A, B, and C for which the frequency of co-transduction determined by two-factor crosses are AB 90%, AC 33% and BC 42%. The values indicate that the B is closer to A than to C. A mathematical formula proposed by Wu is used to relate co-transduction frequencies to map distance, which is C equal to 1 minus D by L whole cube, where C is co-transduction frequency, D is the distance in minutes between two genes, and L is the size of the transducing fragment in minutes. Transformation is a mechanism of genetic transfer between bacteria in which the donor DNA is cell-free DNA in the recipient bacterium's immediate environment. Generalized transduction is the process by which any bacterial gene may be transferred to another bacterium via bacteriophage, and very rarely a small number of phages carry the donor genome. In essence, this is the packaging of the bacterial DNA into a viral envelope. This may occur in two main ways, recombination and headful packaging. Specialized transduction is the process by which a restricted set of bacterial genes is transferred to another bacterium. This type of transduction occurs when the prophage excites imprecisely from the chromosome so that the bacterial genes lying adjacent to the prophage are included in the excised DNA. The excised DNA is then packaged into a new virus particle, which then delivers the DNA to a new bacterium where the donor genes can be inserted in the recipient chromosome or remain in the cytoplasm depending on the nature of the bacteriophage. Co-transduction is a process by which two genetic markers are simultaneously packaged within a bacteriophage for transfer to a new host bacterium. Well, that's all for today's program. I hope you really understood the topic. Thank you.